Hello friends, today we will learn about plant tissue culture media. Culture media are largely responsible for the in vitro growth and morphogenesis of plant tissues. The success of the plant tissue culture depends on the choice of the nutrient medium. In fact, the cells of most plant cells can be grown in culture media. Basically, the plant tissue culture media should contain the same nutrients as required by the whole plant. It may be it may be noted that plants in nature can synthesize their own food material. However, plants growing in vitro are mainly heterotrophic, that is, they cannot synthesize their own food. Let's know about composition of media. The composition of the culture media is primarily dependent on two parameters. First is the particular species of the plant, and second is the type of material used for culture, that is cells, tissues, organs, protoplast. Thus, the composition of a medium is formulated considering the specific requirements of a given culture system. The media used may be solid or liquid medium. The selection of solid or liquid medium is dependent on the better response of the culture. Let's know about the major types of media. First is white medium. White's medium. That is, this is the earliest plant tissue culture media developed for root culture. MS medium. MS means Murasis and Skoog medium. Two scientists named Murasis and Skoog originally formulated a medium to induce organogenesis and regeneration of plants in cultured tissues. These days, MS medium is widely used for many types of culture system. Now come to B5 medium. Its B5 medium is developed by Gamborg. The scientist named Gamborg. B5 medium was originally designed for the cell suspension and cell cultures. At, at present, with certain modifications, this medium is used for protoplast culture. Now come to N6 medium. The scientist named Chu formulated this medium, and it is used for cereal anther culture besides other tissue cultures. Now come to next medium is niche medium. This medium was developed by the scientist named Niche and frequently used for anther cultures. Among the media I told you till now, MS medium is the most frequently used in plant tissue culture work due to its success with several plant species. Now let's know about synthetic and natural media. When a medium is composed of chemically defined components, it is referred to as synthetic medium. On the other hand, if a medium contains chemically undefined compounds, example vegetable extracts, fruit juice extract, plant extracts, it is regarded as natural medium. Let's know about the constituents of media. Many elements are needed for a plant nutrition and their physiological functions and growth. Thus, these elements have to be supplied in the culture medium to support the adequate growth of the culture in vitro. The culture media usually contains some constituents. Let's know about the constituent. First is inorganic nutrients, carbon and energy sources, organic supplements, growth regulators, solidifying agents, pH of medium. Let's know about inorganic nutrients. The inorganic nutrients consist of macronutrients and micronutrients. What is macronutrients? Macronutrients means concentration should be 0.5 to 0.5 millimolar, less greater than 0.5 millimolar per liter, and micronutrient is concentration is should be less than 0.5 micromole per liter. A wide range of mineral salt, that is elements, should apply the macro and micronutrients. The inorganic salts in water undergo dissociation and ionization. Consequently, one type of ion may be contributed by more than one salt. For instance, in MS-medium, K plus ions are contributed by KNO3 and KH2PO4, while NO3 minus ion come from KNO3 and NH4NO3. Macronutrient elements. Let's know about macronutrients element. What are the macronutrient elements? There are six macronutrient elements. First is nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. These are the essential macronutrients for tissue culture. The ideal concentrations of nitrogen and potassium is around 25 millimolar per liter, while for calcium, phosphorus, and sulfur and magnesium it's range between 1 to 3 millimole per liter. For supply of nitrogen in the medium, nitrate and ammonium salts are together used. Now let's come to macronutrients, although their requirement is in minute quantities. Macronutrients are essential for the plant cells and tissues. These include iron, magnesium, zinc, boron, copper, molybdenum among the macro elements. Iron requirement is very critical. Chelated forms of iron and copper are commonly used in culture medium. Let's know about carbon and energy resources. Plant cell and tissues in the culture medium are heterotrophic and therefore are dependent on the external carbon and energy. Carbon for energy. Among the energy sources, sucrose is the most preferred. During the course of sterilization we do sterilization in autoclave. So by sterilization at the in autoclave of the medium, sucrose gets hydrolyzed to glucose and fructose. The plant cells in culture first utilize glucose and then fructose. In fact, glucose or fructose can be directly used in culture media. It may be noted that for energy supply, glucose is as efficient as sucrose, while fructose is less efficient. Let's know about organic supplements. The organic supplements include vitamins, amino acids, organic extract, activated charcoal, and antibiotics. Vitamins. Plant cells and tissues in culture, like in natural plants, are capable of synthesizing vitamins, but in suboptimal quantities. Adequate of support growth. Therefore, the medium should be supplemented with vitamins to achieve good growth of cells. The vitamins added to the media include thiamine, riboflavin, niacin, pyridoxine, folic acid, pantothenic acid, biotin, ascorbic acid, myoinositol, para-amino benzoic acid, and vitamin E. Now let's know about amino acids. Although the cultured plant cells can synthesize amino acids to a certain extent, media supplemented with amino acids stimulated stimulate cell growth and help in establishment of cells line. Further, organic nitrogen like it's in the form of L-glutamine, L-aspargine, L-arginine, and L-cytosine. Cysteine, sorry, not cytosine, that is cysteine. L-cysteine is more readily taken up than inorganic nitrogen by the plant cells. Now come to organic acids. Addition of Krebs cycle intermediates such as citrate, malate, succinate, or fumarate allow the growth of plant cells. Pyruvate also enhances the growth of cultured cells. Now come to organic extracts. It has been a practice to supplement culture media with organic extracts such as yeast, casein hydrolysate, coconut milk, organic juice, tomato juice, and potato extract. It is, however, preferable to avoid the use of natural extracts due to high variations in the quality and quantity of growth promoting factors in them. In recent years, natural extracts have been replaced by specific organic compounds. Example: replacement of yeast extract by L-asparagine, replacement of fruit extracts by L-glutamine. Thank you.